Hello dear students, welcome back again to Chinmoy's YouTube channel. So yesterday we have already given the introduction of bacteria. We have already started about the shape, size, structure, about their reproduction. We have already discussed about the respiration and how the binary fission method is happening and how the endospore formation is happening. So yesterday we have already done that. So today we are here with the economic importance of bacteria, that is how they are useful as well as what are their harmful effects. So without any delay, let us start with today's episode of bacteria. So what are the learning objectives of today's lesson? So at the end of this lesson, we will be knowing that what is the useful role of bacteria as well as what are the harmful role of bacteria. So if you want to know this in details with diagram, with proper description and notes, you need to subscribe to my channel. So for knowing more updates on your 9th biology lessons as per ICSE curriculum, please subscribe to my channel and don't forget to like and share my videos. And if you are subscribing to my channel, the regular updates which I am posting, you will get that, and you will follow my lessons in a systematic manner in the way I am giving you the lessons as per your curriculum. So stay tuned and subscribe to my channel. So let us start with today's episode of bacteria and fungi. Now here, when we are starting with the useful role of bacteria, we will take the useful role of bacteria in agriculture. So in agriculture, when we are taking this, first we should talk about the process of nitrification. So what is it? This is very important. Now plants, they are getting the nitrogen from the soil. Now for synthesizing certain proteins. So they are absorbing the nitrogen from the soils for synthesizing certain proteins. Now they are absorbing these proteins in the form of nitrates. Okay, so they are absorbing these proteins in the form of nitrates. So how they are absorbing these proteins in the form of nitrates? Because the process of conversion of ammonia into nitrites and nitrates. So in the soil, that the nitrogen is present in the form of ammonia. Okay, so through manure, through fertilizers, or through the waste of animals, they are present in the form of ammonia in the soil. So they are present in the form of ammonia in the soil. Okay, now when they are present in the form of ammonia in the soil, what they are being done? So they are being converted to nitrites and nitrates. Okay, so they are being converted to nitrites and nitrates, and this process is called nitrification and is brought about by nitrifying bacteria present in the soil. So this is the role of nitrifying bacteria, to convert ammonia into nitrites and nitrates. Okay, so there are some bacteria which are converting this nitrogenous waste of animals or in dead bodies of plants and animals to ammonia. Okay, so few important bacteria, they are nitrosomonas is a very important bacteria which is oxidizing the ammonia to nitrates. Okay, and you can say the nitrobacter then oxidize the nitrites to nitrates. Okay, this nitrate is taken up by the plants and is used in several biochemical process. Okay, so this is all is happening. So the conversion of this ammonia into nitrates and nitrites is done by several bacteria. So the name of the bacteria, common bacteria, is nitrosomonas and nitrobacter. These are the bacteria which are converting ammonia to nitrates, and nitrobacter is a bacteria which is oxidizing nitrites to nitrates. Okay, is it clear? So nitrites are NO2 and nitrate is NO3. Okay, so is it clear? They are oxidizing it. They are oxidizing nitrites to nitrates. Okay, so let us move on to the next part. Now here comes the next process, which is a useful role of bacteria in agriculture, is nitrogen fixation. So what is happening here? The process of conversion of the free atmospheric nitrogen. So nitrogen is always free in atmosphere. So it is converted into nitrogen compounds. It's called nitrogen fixation. So the free living nitrogen, what is the nitrogen fixing bacteria like Azotobacter and Clostridium, or these are the symbiotic bacteria. So what is the meaning of symbiotic? So when they are in close association with each other, so they are symbiotic. So one is helping another to do or perform a work. So these symbiotic bacteria which live in the root nodule, so these bacteria Azotobacter and Clostridium, they are in the present in the root nodules of leguminous plants. Okay, So this name of leguminous plant is rhizobium, which helps to fix the free atmospheric nitrogen to nitrates. So the atmospheric nitrogen, free atmospheric nitrogen is being converted to nitrates with the help of this nitrogen fixing bacteria. So with the help of this nitrogen fixing bacteria, so whose name are Azotobacter and Clostridium. So these are the, if a question comes that name two nitrogen fixing bacteria, so you can name as Azotobacter and Clostridium. Okay, so these are present in the root nodules and they help in the conversion of free nitrogen to nitrates. Okay, so let us see what is here. Now here you can see this is a cycle, atmospheric nitrogen. This is being converted by the help of nitrogen fixing prokaryotes or bacteria to ammonia. So again nitrifying prokaryotes. So this is present in the root nodules of leguminous plants. So these are the root nodules of leguminous plants. Again this ammonia is converted to nitrites. So this is again converted to nitrates, and again these compounds are converted to atmospheric nitrogen by denitrifying bacteria. Okay, so this is a continuous cycle. This is a nitrogen cycle which continuously goes on between the atmospheric nitrogen, between the soil, bacteria, and again to the atmosphere. So this is rather a cyclical process. So atmospheric nitrogen is being fixed in by the nitrogen fixing bacteria here, that is Azotobacter and Clostridium, which is again converted to nitrates by, we have told in the earlier slide only, we have told that Nitrosomonas and this Nitrobacter are the two bacteria which are converting these nitrates to nitrites. So these are oxidized by Nitrobacter and Nitrosomonas to oxides, that is nitrates. And then from these compounds, again with the help of denitrifying bacteria, we will tell that in the next slide only, they are converted to atmospheric nitrogen. So that is the denitrification process. So is it clear? This one, nitrogen fixation, that is fixation of nitrogen to nitrogenous compounds, is called nitrogen fixation, and it is mainly happening with the help of Azotobacter and Clostridium. Now here comes the third process of useful effect of bacteria in agriculture, is the denitrification. So denitrification is what? It is the degradation of the nitrate to the nitrite salts to the from nitrate and nitrite salts to elemental nitrogen, that is N2. So NO2 and NO3 are being converted to elemental nitrogen, that is atmospheric nitrogen they are being converted to, and that process is called denitrification, and it is carried out in the soil by free living bacteria called Pseudomonas. So the first process, that is the first process of nitrification, was being held by the bacteria Nitrosomonas and Nitrobacter. The second one, or the nitrogen fixation, was brought about by Azotobacter and Clostridium, and the third one, or the denitrification, is happening with the help of Pseudomonas. Just remember the names of these bacteria in your mind, so that you you should not mix up one with another one. Okay, so Pseudomonas is a denitrifying bacteria, nitrogen fixing bacteria are Azotobacter and Clostridium, and Nitrosomonas and Nitrobacter are the nitrifying bacteria. Okay, so you should remember that you should not mix them up. Okay, so let us see the diagram. Now here the same thing, this ammonia and nitrate, they are converted, their nitrification is happening, oxidation is happening here, and again these things, they are being converted by denitrification process to elemental nitrogen. So these, in the root nodules, we have told earlier also, there are nitrogen fixing bacteria like Azotobacter, and you can say that Azotobacter and Clostridium, they are fixing the nitrogen in the form of nitrates and nitrites, and that is again converted to elemental nitrogen with the help of Pseudomonas, which is a free living bacteria and which is carried out in the soil only. So this is the process of denitrification. So now we will move on to the next process. Now here we will be seeing all the combination of all the processes we have studied earlier, that is nitrification, nitrogen fixation and denitrification process together in this nitrogen cycle. So let us see first what is happening. This is atmospheric nitrogen. So this rabbit is inhaling that atmospheric nitrogen. It is passing out stool, so that is a decomposed or decaying matter from the body. It is passing out, so it is mixing in the soil. So soil is having several decomposers like aerobic and anaerobic bacteria as well as fungi. So the nitrogen fixation happens in the soil also, so that is converting it into ammonia. Okay, so ammonium with the help of nitrifying bacteria, it is being converted into nitrites, NO2. Then nitrites with the help of again this nitrifying bacteria, they are converting or oxidizing nitrites to nitrates. Okay, with the help of nitrifying bacteria, these are being converted into nitrates. Then again, by the presence of denitrifying bacteria like Pseudomonas and all, they are being converted to the atmospheric elemental nitrogen. So this is a full cycle which is happening. So it is first from the atmospheric nitrogen by nitrogen fixing bacteria which are present in the root nodules. They are present in the root nodules and they convert it into, by the process of ammonification, they are converting it into ammonium, and that is being again converted to nitrites, that is being converted by nitrifying bacteria to nitrates, and with the help of denitrifying bacteria again back to elemental atmospheric nitrogen. So this is the nitrogen cycle which keeps on going as a continuous process. So this is, if you remember this flow diagram only, it is very easy for you to remember the process of nitrifying bacteria and denitrifying process as well as this nitrification. So only the name of the bacteria you should remember. Okay, so let us move on to the next one. Now these two things we should know, that is what is decay and putrefaction. So what is decay? Decay actually it is the complete breakdown of the organic matter by bacteria without giving out foul smell. Now what is putrefaction? That is the incomplete breakdown of the organic matter by bacteria during the process, and a foul smell is always given out. Now bacteria, they are always scavengers in nature and they are playing an important role in recycling of the several nutrients of the nature. So bacteria helps in generating biogas. So how biogas is generated? It is acting on the waste products of human waste, cattle, dung, garbage, etc. and it is turning them into biogas, which helps in the regeneration of energy, and you can use biogas in many other ways also. So this is how we can tell that bacteria has several other uses also. It is helping in the production of biogas, and decay and putrefaction are the two important terms that you should just know. What is decay? Decay is the complete breakdown of organic matter and there is no foul smell given out, whereas putrefaction is the incomplete breakdown of the organic matter by releasing a foul smell. So these are the important facts you should just know for your knowledge. Okay. Now next, how you are using bacteria in industry. So first one was in agriculture. Now next is use in industry. Now curd is prepared by fermentation. We know that curd is prepared by fermentation of milk sugar in the milk into lactic acid. So with the help of what? With the help of lactobacillus in the industry we get curd by this process only. The milk sugar is converted into lactic acid, okay, from the milk by the help of lactobacillus bacteria. Now vinegar production is also used, bacteria is used where the ethyl alcohol, it is converted into acetic acid or vinegar, okay, by the action of acetobacter and bacillus. Okay, because ethyl alcohol, conversion of ethyl alcohol to acetic acid, which is vinegar, it is also done by the use of bacteria, and the name of the bacteria are acetobacter and bacillus. So these are all the processes or the uses of bacteria in industry. In large scale, bacteria is used to produce curd, to produce vinegar, and this retting of fibers of jute and hemp. So what is retting of fibers? It is actually the extraction of the original fibers from the raw one. So there also microorganisms are used, that is bacteria is used to extract the pure form of jute from the raw fibers. Now bacteria, another use is that it is added to the tea or coffee to get the flavor. So how is this process is done? This is called the curing of the tea. So what is curing of tea? Now in this process, bacteria, it eats away the unwanted compounds, produces the characteristic flavor. So whatever unpleasant or unwanted compound is present in tea, that is being eaten away by the bacteria which is added to the tea, and it produces a characteristic flavor. So we get the actual flavor of the tea or coffee which we want to get, so which is the ideal flavor. So bacteria is also used in tanning of leather, as the raw leather is brittle and it contains fatty acids, fatty compounds, okay. So these fatty compounds, what they do, they are actually, when raw skin is taken, it is to be tanned, is immersed in water containing bacteria. So for this tanning process also of the leather, we can say that bacteria is used, which is removing all the fatty compounds and making the skin soft. So it is removing all the fatty compounds and making the skin very, very soft. And then only these leather can be used for making bags, for making any other purposes. Okay, so in industry, the leather which is raw, it is very stiff, very brittle because of the presence of some fatty compounds which has to be dissolved when they are immersed in water with with bacteria. Okay, so these are dissolving this fatty compounds and making the skin soft for industrial use. Okay, then you can use it for making sofas, for making leather jackets, for making whatever you are using, shoes, bags, etc, etc, etc. Okay, so now specific humidity and temperature is always required and maintained for this tanning purpose. Okay, this should be maintained in the industry, otherwise this tanning process will not happen. Now here, use of bacteria in medicine. So production of vitamins is very important. For many bacteria, they are actually present in the intestine of the humans, and they synthesize certain vitamins such as vitamin B complex. So this is gut microbes actively synthesize and remodel vitamin B12. So this is just a model I have taken. So they are synthesizing vitamin B complex in our intestine of humans. So in some vitamins like live in the intestine of herbivorous animal and help in the digestion of cellulose. So many other herbivorous animals like a cow and all, their also intestine has certain amounts of these bacteria help in the production of some vitamins which help to digest the cellulose. Okay, so because they are chewing the plants, they are staying on plants and all, so when they are chewing the plants, the cell wall is there in the plant cell, right? So cell wall is made up of cellulose. So we all know that cell wall is made up of cellulose. So this needs to be digested when the animals are ingesting those leaves and all from the plants. So this digestion of cellulose is also being done with the help of certain bacteria. So that bacteria is present in the intestine of some herbivorous animals. Okay, now and human intestine, we are already producing some vitamin B complex. This vitamin is being produced with the help of bacteria only. So gut bacteria helps in the production of vitamin B complex. Now here comes the production of medicines. So what will happen here now? Antibiotic drugs are there which are being formed in the factory due to the production of many antibiotic drugs. We are getting cured from many types of diseases nowadays. So what are the antibiotics? Mainly mention may be made of streptomycin, aureomycin, terramycin, penicillin. These are very common antibiotic drugs. There are many more antibiotic drugs, but we mention should be made of very few because we can't learn or know all the names here. Just 4 or 5 I am mentioning. You can go through many other antibiotics also. Now genetically induced bacteria produce insulin, which is also used in research laboratories and drug production. So genetically induced in lab also insulin is prepared with the help of some bacteria only. Okay. Now just quickly we will tell what is antibiotic and how it is produced. Now what is antibiotic? Antibiotic is a chemical substance which is produced by a living organism which can prevent the growth of some disease causing bacteria, is called an antibiotic. So it is preventing the growth of some disease causing bacteria and it is known as antibiotic. So antibiotic was first discovered in 1929 by Sir Alexander Fleming, and the term antibiotic was first coined by Selman Watson in 1942. So in 1929 by Sir Alexander Fleming, penicillin was the first antibiotic which was discovered, and the term antibiotic was coined first by Selman Watson in 1942. Now Penicillium notatum was the first mold from which penicillin was obtained. So Penicillium notatum, just you should remember these terms. Penicillium notatum is the bacteria or the first mold from which penicillin was obtained. Okay, so we can say that commercially penicillin is produced from Penicillium chrysogenum, a fungus. So it was first commercially derived from Penicillium chrysogenum. So penicillin was the first antibiotic discovered, and Penicillium notatum was the first mold, and it was first discovered from fungus, and as well as it was synthetically produced, commercial or synthetically produced. Now there are production of few more antibiotics. We can say that streptomycin is one of the most widely used antibiotic which is formed from Streptomyces gracius. Now, chlorotetracycline is produced from the bacterium Streptomyces aureofaciens. So, these scientific names you should remember, you should so that you can recall that if they are giving you the name of this antibiotic, you should write from which bacterium, that is Streptomyces aureofaciens, from this bacteria it has been derived. So, chloramphenicol is produced from a bacterium Streptomyces venezuelae. Okay, so these are the from these bacteria only these antibiotics have been derived, and streptomycin is one of the most widely derived antibiotic from Streptomyces gracious. Okay, so now we will move on to the we can say that use of antibiotics. So what are the use of antibiotics? Now we can say that they are actually used to fight diseases. So till now we have been telling that they are used to fight diseases. So what are antibiotics being doing? So they are used in the preservatives as you can use antibiotics for the preservatives of meat and fish. Okay, for preserving meat and fish we can use. Now we can also use antibiotics to treat animal from several infections. So we can treat animals also. We can treat animals to prevent certain infections and well, because whenever we are getting infected through any bacterial diseases, doctors are prescribing us with antibiotics. So whether it is eye infection, nose infection, ear infection, stomach infection, we are being given doses of antibiotics for 5 to 7 days to treat this infection. So let us move on to the next. Now next comes serum. So what is serum? Serum is actually a clear yellowish fluid. After separating, we all know that after separating the blood into its solid and liquid components, after it is allowed to clot, the clear yellowish fluid is called the serum. So this serum contains the antitoxin part. So the clear yellowish part, after the blood coagulation is done, so the liquid part which is yellowish in color is the serum, and serum is injected into a body to build up resistance. So because they are having antitoxins in them, they are inserted into the body to build resistance against certain microorganisms and their toxins before the microbes actually enter the body. So they are acting to build the immunity, to build the resistance of the body before the toxin enters the body. And it is easy to neutralize and kill the bacteria when the serum is being injected in the body. Okay, so when the serum is already been injected in the body, it is containing the antitoxin, and this helps to neutralize and kill the bacteria. So let us see the process here. Now how it is formed, how the preparing of serum is done, let us see here how it is prepared. Now for preparing serum, what we are doing, a small dose of bacterial toxin is introduced into the blood of a healthy animal such as horse. Suppose it is injected with such a bacterial toxin. Okay, so then the body is producing antitoxin. Here antitoxins are being produced. So then the blood of the animal is again taken out. Okay, and it's separated in this separation in this machine. The separation is done of the solid and the liquid serum part. Now the separation of antibodies is done. Then the antibodies, this concentrated antibodies is taken out, and this formulation is done, which is being injected into the body. So now nowadays we have used or modified bacteria by introducing human genes in the E. coli or Escherichia coli, and we have extracted such bacteria, that is the hormone insulin was we can say that was the first such substance which was produced by E. coli bacteria. So is this process clear? Now preparing of serum, it's being injected, the animal is being immunized, the blood is drawn out, and the serum and the solid components, blood cells are separated. Then separation of antibodies, the antibodies are taken in concentrated forms and used in the form of medicines or formulation. Is it clear? Now let us move on to the next one. Now very few examples of genetically modified bacterial serum I have taken. So just learn them. First one is insulin from E. coli. Second one is the blood coagulating factor 8 for the treatment of hemophilia A, and factor 9 for the treatment of hemophilia B. Now just you remember these things, might be in your exams. Some few questions might come from this. Name some genetically modified bacterial serum. So then you can mention any one of these three. Okay. So actually we are reading this lesson in a bit detail. So if you want to answer any questions from any part, you can get the question and you can answer it properly. So I am doing a detailed description of this bacteria, that is uses and harmful effects also we are going to discuss. So if you have not yet subscribed, please do subscribe to my channel to know all the lessons in detail. So let us move on to the next one. Now we have come here to vaccine. So what is a vaccine? A vaccine is actually a preparation of live weakened germ used to produce active immunity against a disease. So it is a live germ only, live bacteria only, which is weakened or heat killed actually, to use active immunity against that disease. So this name was first coined by Edward Jenner, who first coined the name vaccine. Now we can say that typhoid vaccine has been given to kill the bacteria, then BCG vaccine for tuberculosis, and these are all live weakened vaccines. So nowadays this live weakened bacteria, we have again taken the vaccine for COVID-19, right? So we all have been given double doses of COVID-19 vaccine against this disease also. This is a viral infection, though vaccines have been produced. So in the similar way only, this is a bacterial vaccine which is produced from bacteria, and this what we have taken COVID vaccine is against the viral infection, that is COVID-19. Okay, so there we have taken the live weakened part of the virus which have been used as a vaccine form. Now, after so many useful effects of bacteria, at last we have come to the harmful effects of bacteria. Now we will quickly jot down that. Now, some common diseases caused by bacteria are diphtheria, typhoid, tuberculosis, leprosy, cholera, tetanus, dysentery, diarrhea, gonorrhea, and botulism. So many mention more may be made. So in animals, bacteria causing anthrax, like swelling in the body, and low milk yield. So these are the bacteria which is infecting the anthrax, or the anthrax disease, which is caused by the bacteria. Okay, now plant diseases like leaf spot disease in cotton plant, which is caused by Xanthomonas malvacearum. Okay, so this plant diseases like leaf spot disease in cotton, bacterial wilt in potato, which is also caused by Xanthomonas solanacearum. So these are same group bacteria. So that is Xanthomonas malvacearum. This is Xanthomonas solanacearum. So just remember these scientific names of these bacteria. And there is gall formation in plants also by Agrobacterium tumefaciens. So Agrobacterium tumefaciens is causing gall formation in plants. Now next we will move on to the last slide. Now how another harmful effect of bacteria is spoiling the food. So how it is spoiling the food? It is spoiling meat, fish, and other items. It actually it's releasing toxin into the spoiled food, and it's causing food poisoning. Botulism is a kind of food poisoning. Bacteria is also used as bioweapons to kill millions, lakhs and lakhs of people. So it's used as a bioweapon also, so that they can generally spread among the in the form of disease among the people, among the animals, to kill a huge amount of human population as well as animal population also. So that like that only they are used as bioweapons. Now Bacillus anthracis is a bacteria which it is producing anthrax causing toxin. Now anthrax is a deadly disease. Generally this anthrax bacteria, they produce cell like spores, and that allow them to live in the dormant state in the soil. So when they're used as a weapon, what they do, they enter the body, and there in the immune system, they destroy the immune system. There the spores become active, and they keep on reproducing in large numbers inside the human body. So they start reproducing inside the host body. They produce toxin, and that causes the anthrax disease, and which is a deadly disease. This is how these bioweapons are being introduced in human body, in animal body, so that as soon as they enter the body, they destroy the immune system, the spores become active, and they start reproducing in huge numbers in the host body. Now thus the toxin is causing the disease in the body, and it is killing a huge population of animals as well as humans. So this is all in today's episode of useful and harmful effects of bacteria in human life. So this is all I have completed today. And I know you have all gone through this episode, and if you have not yet subscribed to my channel, please do subscribe to my channel, because in the next episode I'll be back with fungus and its economical importance, as well as the shape, size, reproduction and structure of the fungi. So stay tuned, stay updated with all my videos. Thank you.